Hi, my name is Stephen McGee. I am one of the bioinformatics specialists here at the Greenwood Genetic Center. I've been with the GGC for over 7 years now, developing and implementing our genetic sequencing pipeline for treatment of our patients who suffer from a variety of genetic disorders. As part of my daily routine, often I will take genetic sequencing data from a patient affected with a genetic disorder and compare them against family members who may be unaffected. In a situation where a child affected with a disorder is compared with two unaffected parents, this method will allow us to subtract out variants or mutations which are shared between the child and either of the parents. We can then focus our attention on mutations which are unique to the child and determine whether or not any of these are causative of the genetic disorder itself. We perform these analyses using software which we have developed or through free, publicly available online databases. We then report out our findings to directors and physicians who will then use our results to determine the next course of action in treating the affected patients. As a discipline, bioinformatics really gained headway after the successful completion of the Human Genome Project in 2003. Then it was publicly stated that the human genome had been successfully sequenced. A common misconception in this statement is that the DNA sequence for all human beings was known, whereas in reality, the DNA sequence was known for only a handful of volunteers who were recruited by the directors of the initial project. In fact, the DNA sequence for each and every individual human being is quite unique. We, as bioinformaticians, acknowledge this uniqueness and attempt to find patterns within the genome to understand the sheer complexity that lies within. Here at the GGC, most of my efforts are focused on analyzing the coding sequences of the genome, which encompass approximately 20 to 30,000 known genes. These regions, referred to as the exome, represent only 1% of all DNA in the human body. Though my analyses focus on this meager 1%, the remaining 99 are by no means to be considered junk DNA. Research is ongoing as to the nature of the functionality of these intronic and intergenic regions. Two of the requirements for being a bioinformatician involve a thorough understanding of the concepts of biology as well as computer science. Our team is thoroughly experienced in a variety of different programming languages as well as many different topics in the field of biology, all of which we learned while in school. Personally, I attended the University of South Carolina at Columbia where I received my bachelor's degree. Afterwards, I attended the University of North Carolina in Charlotte where I received my master's in bioinformatics while studying microarray technology. Currently, I am working towards my doctorate in finding novel ways to incorporate artificial intelligence in deriving patterns from the human genome. With the discoveries that have been made in the past 14 years since the successful completion of the Human Genome Project, we are seeing the development of revolutionary new patient treatments and thereby witnessing the advent of personalized medicine itself. We are living in the golden age of biology, and there has never been a better time to be a bioinformatician.